Good evening, everyone. Just let me know, are you guys listening? Yes, thank you. So, good evening, and welcome to the second and the final day of our third international webinar on Productions Challenge. So, first of all, I want to thank you everyone for the presence. It's very gratificating for GCA to have the participants in the event in both days, today and yesterday. And in additionally, I want to express our deep gratitude to our speakers for their time and for sharing their knowledge with us. We are very honored to be by your presence. So, first of all, we are going to have our first speaker, that is Ana Luiza de Souza. She's going to, so first I'm going to be giving a little introduce about Ana. So Ana has a bachelor degree in biological science from Lavras, and a PhD in entomology from Federal University of Lavras. She completed her postdoc at Rutgers University of New Jersey. Currently, Ana is in entomology science at Syngenta Seeds Red Innovation Center in Malta, Illinois. She will share her experience on the challenge in controlling insect pests in blueberry farms. We are confident that today we have a productive discussion and valuable insights. So, without further ado, let's start our event. It's a great pleasure that I invite Ana Luiza to start her presentation on challenge in controlling insect pests in blueberry farms. Ana, the floor is yours.
Thank you, Yuki. Hi, everyone. Good evening. So, everybody can hear me, right? So I hope so. So first of all, I would like to thank you, Professor Raquel Pires and Yuki, for the invitation for attending this webinar as a speaker. And it's such a pleasure to be here today and share some information with all of you. Okay, so I'm gonna share my screen here. Can everybody see my screen? Yuki, can you please confirm if you guys are seeing my screen? Okay, so for today, I'm going to talk about the challenges in controlling insect pests and blueberry farms. So the topics of these presentations will consist in the overview of the blueberry crop importance in the United States, the challenges for blueberry farmers, the researchers, the outcome, and the challenges. Again, okay? So let's start the first topic that is about the overview of the blueberry crop importance in the United States. So the blueberries, or as we know in Brazil as mirtilo, they are native to North America, and they have a common name here that is a high bush blueberry. It is common name refers to the relatively tall structure, as you can see on this picture in this slide. So the blueberries, they are also part of traditional cuisines and modern food trends. It is a significant crop in the United States, and the blueberries are a significant crop with a growing importance in both economic and health contexts. So regarding the economic context, so the growers of United States high bush blueberries, they generate more than $4 million in annual economic impact. Translating this to more than $12 million flowing into the United States economy every day of the year. Also, the blueberry industry creates and sustains more than 44,000 full-time equivalent jobs each year. And the United States is the world largest producer of blueberries. So here in this map, we can see the top blueberry producing states in the United States. So we have Washington, Oregon, California, Texas, Florida, Georgia, North Carolina, New Jersey, Michigan, and Minnesota. Those are the top blueberry producing states. So the blueberries, they also contribute to crop diversity in the United States agriculture, and they support pollinator populations, particular bees. So the ongoing research into new varieties for improved yield and disease resistance, and also in studies on sustainable farming and practice and pest management. So the United States export blueberries to various countries, contributing to agriculture trade. So besides all this contribution of the blueberries, they are also important for our health. So what are the health benefits of the blueberries? They are rich in antioxidants, particularly anthocyanins. They are high in vitamin C, fiber, and manganese, and they are associated with improved heart health, brain function, and blood sugar control. So, if you wanted to be a blueberry grower, you had to consider all the requirements. So, what are the best conditions for growing high bush blueberries? And I would say that the blueberries, they are a kind of peak in the requirements. Okay. So the first condition is the soil pH. The soil pH that the blueberry plants they like is acidic soil with pH between 4.5 and 5.5. Yes, it's very low. The other topic is the soil type. The soil needs to be very well drained and rich in organic matter. And the ideal soil types are sand loam or peat-based soils. So regarding the sunlight, the area of the crop needs to have 6 to 8 hours of full sun daily for best fruit production. They can tolerate partial shade, but this may reduce yield. And no one wants to reduce the yield, right? Regarding the water, we have to have a consistent moisture and a well-drained soil to prevent root rot. And what about the temperature, Anna? What are the ideal temperature? to grow blueberries. So the ideal temperature range for grow and fruit maturation is 20 to 30 degrees Celsius. But when the plant is on the vegetative stage, they can support minus 13 degrees Fahrenheit, that means minus 25 degrees Celsius, during the winter season. This is the time when the plants, we can say that the plants are dormant. Okay? So humidity. So they required moderate humidity levels, and they have to have a good air circulation to prevent fungal diseases. The soil nutrients. The soil needs to be in low nitrogen, and we have to use a balanced acid forming fertilizers. What about the spacing between the plants and the spacing between the rows? Between plants needs to be 4 to 5 feet, and between rows needs to be 8 to 10 feet. Pollination. Pollination for a blueberry, it is very critical and important. So we have to plant at least 2 varieties for cross-pollination, and we have to encourage bee activities for better fruit set. So after the harvest, what we need to do, what are the conditions after the harvesting? The pruning. Annual pruning to maintain shape and encourage new growth. Mulching. So here in these pictures, you can see the mulching in the top of the soil. It's recommended 2 to 4 inches of organic mulch to retain moisture and suppress weeds close to the plants. And wind protection. So shelter from strong winds, especially in colder climates. So what does that mean? So basically, all the blueberry farms here in the United States, they are surrounded by forests. So the forests, we have high trees. So that means we have wind protection with through this forest, through these high trees. Okay? And the soil depth. The soil needs to be at least 18 to 24 inches of good soil for root development. Okay? So, yeah, we have the overview of the blueberry crop in the United States. We have the requirements to grow the blueberries in the United States. But as the same for the other crops, the blueberry crops, they have challenges. And what are the major challenges faced in blueberry farming? So the first one is the soil management. So the growers, they need to maintain an optimal soil pH and the fertility. The second point, the second challenge is the pest and disease control. The third one is the climate and weather risks. This involves frost damage during flowering. If you have a frost during the flowering, the flowers will die, they will fall. Heat stress during fruit development. If we have heat stress during fruit development, the fruits will fall. and drought or excessive rainfall. The pollination. This involves ensuring adequate pollination for good fruit set, managing bee populations, or hunting hives. So this pollination is a really challenging right now because pollination is related to pest and disease controls, because I'm going to talk about this further, but since it's really challenging to control the insect pests and they apply a lot of insecticides, this also affects pollination. So the other point is harvest timing and labor. So determining optimal harvest time for quality and yield, securing skilled labor and hand harvest. Yes, the blueberries are hand harvests, as you can see in these pictures. So the other major challenges, post-harvest handling. So they have to have a proper cooling and storage to maintain fruit quality, managing shelf life for fresh market sales. Because of course, since they are having the final product as a fruit, so they will commercialize this product. So they have um, to take care of the proper cooling and storage to maintain the fruit quality. Because if they do not do this, when the fruit arrives to the, for example, in a grocery store, people are going to start complaining because they will not have a good quality. The other challenge is the market competition. So competing with imports from other countries and meeting consumer demands for quality and organic production. Water manager. So blueberry farms need to have an efficient irrigation systems, and this is very expensive. Variety selection. So it's very challenging to choose cultivars that were suited to local climate and market demands, involving balancing flavor, size, and productivity. And the last one, the regulatory compliance, adhering to food safety regulations like insecticide applications and meeting organic certification standards for the organic farmers. So all those challenges require ongoing management and adaptation to ensure a successful blueberry production. Farmers often need to stay updated on the latest research and technology to address these issues effectively. So, we're going to talk more about the challenging that involve pest control. Okay, so since I'm an entomologist, I'm gonna focus on the insect pest control and not in the disease control. Okay, so thinking about blueberry farms. So what are the insect pests that commonly affect the blueberry farms here in the United States? The number one, and we have a similar problem in Brazil with in fruits. So is the spotted winged Drosophila suzukii. This is a highly invasive pest. The females lay their eggs in the ripening fruits, causing significant damage. And this difficult to control, and this pest is difficult to control due to the rapid reproduction. So the spotted winged Drosophila, they have multiple generations in a year. The second one is the plancurculio, Conotrachelus nenuphr. That's here is in this picture. The adults it feeds on the development fruit and the larvae, and the females they lay their eggs in the fruits, and the larvae cause the fruit to drop. We also have other pests: blueberry maggot, Japanese beetle, blueberry gall midge, blueberry bud mites, and blueberry steam butter. I'm not gonna go in details about the other pests because for today's presentation, I'm gonna focus in the two first ones, the spotted wing Drosophila and the plancurculio. Okay. So, what are the challenges in managing insect pests in blueberry farms? So we have a list of these challenges here, and let's go over them a little bit, okay? The first one, so as we can see here in this slide, we have a diverse pest types. So dealing with various insect pests at the same time, and each one require a unique strategy. And I'm going to talk about that involving a plancurculio and a spotted wing Drosophila suzukii. Okay. The second challenge, resistance issues. The overuse of pesticides can lead to resistance, affecting their effectiveness. Yes, we are having resistance issues already in blueberry farms, especially for spotted wing Drosophila. Okay. Environmental impact. Pesticides may harm beneficial insects, such as bees for pollination and natural enemies to help control these insects, and disrupt the ecosystems. The other challenge, the weather variability. Changing weather conditions can affect pest populations and control efforts. Yes. Regulatory limits. Compliance with pesticides regulations can restrict available options. The other topic, complexity of integrated pest management. Implemented in IPM involves coordinating multiple control methods. It's not only one method. And not two, involves many. Okay. Monitoring demands. Effective controls requires regular monitoring and precise timing, which can be labor intensive. And financial constraints. Budget limitations may restrict access to advanced pest control technology. So there is a lot of challenges in blueberry farms, right? So what the growers do, how they going to overcome these issues. So basically what happened is that here in the United States, we have a really big and strong connections between growers and universities. So why? We have a really strong extension program in the universities. Okay. So the challenges in controlling insect pests, this is an issue for organic and conventional farmers. So the growers, they seek help and they seek help through the extension programs. So the extension programs, we have a really close and a really real contact with the growers where they bring to us the issues they are facing and they seek help, they seek guidance. And the growers, they have a very open mind where they will allow us to do the research in their farmers. And this helps a lot. So first, so how this works. So the growers, they come to the university where we have the extension presentations and extension representatives, and they're gonna expose the issues that they are facing. So you're gonna have a lot of meetings, and then we are gonna develop a project, and we are going to present this project to the growers. So after that, we have to apply these projects to get the funding. And once we get the funding, we can start the research. Okay? So basically, what the growers are seeking, there are more options involving research in biological control and, of course, in breeding. But today, I'll be focused on the research in biological control. Okay? And you guys will everybody will understand um, why I'm gonna talk about this research. Okay? So the that I'm gonna share with everybody, this was a research that I conducted during my postdoc in New Jersey at Hawkers University. And so at that time, the organic growers, they came to us seeking for help to control the plancuculio. This is the plancuculio is a coleoptera, and so I developed a project. We applied for fundings. We got the fundings, and I developed the research. So I'll be sharing here the details about this research, okay, to help the blueberry organic growers. So the title of the research was Entomopathogenic Nematodes for the Management of Plancurculio, Coleoptera curculionidae, larvae and pupae in high bush blueberries. So I conduct this research and I have the support of all those people here. Okay. So let's go to a quick introduction. So the Plancuculio conotrachelus nenupor is a key pest of stone and pond fruits, such as plums, peaches, apples in the United States. And in New Jersey, it is the one of the most important fruit feed pests of commercially cultivated blueberries. So the Plancurculio adults, they move from the overwintered sites to the blueberry sites in spring around April and May. So the adults will start feeding, mating, and the females will lay their eggs in blueberry fruits, where the larvae develop inside the fruit in the first, second, third, and fourth instar. Okay? So the larval feeding causes fruit develop a prematurity and fall off the brush. So the mature larvae, the fourth insta larvae, they exit the fallen fruit that was a bee on the ground, right? And they will burrow into the tissue pupate. So the pupae, we can find the pupae in the soil, okay? And it takes around 30 days for the adults to emerge. So this is the life cycle of the plancurculio. So the main management tactic used to suppress plancurculio adult, it was being above ground applications of insecticides. But these involve environmental and regulatory concerns, right? And approach needs to be developed to control the plancurculio and reduce the amount of insecticide used. So where the growers, they came to us seeking for help, and they were saying that they were interested in biological control. So the use of entomopathogenic nematodes are one potential option to control plancurculio. Okay, and there are some research that where they tested the application of entomopathogenic nematode species, targeting plancurculio larvae in soil in some crops such apple, peach, and plums. However, at that time, there were no entomopathogenic nematodes were tested for the control of plancurculio and high bush blueberries. So based on this information, this project, this research, have 3 objectives. And we decided we need to have, since we don't have information regarding laboratory for plancurculio, entomopathogenic nematodes and plancurculio, we need to test this in laboratory. And based on the results from the laboratory, we can go to the field. So this is experiment, they were done in 2 years, in 2020 and 2021. Okay? So the first objective was to determine the persistence of 4 entomopathogenic nematodes species: Sternenema riobravi, Carpocapsi, feltii, and Heterohabititis bacteriophora in a seed blueberry soil. Why this objective? Because since blueberry soil is acidic, we didn't know if the nematodes would persist in this soil. What that means, would survive in the soil. Okay. The second objective was to compare the virulence of these EPNs to plancurculio larvae and pupae. What does that mean? Will these entomopathogenic nematodes be able to infect larvae and pupae of plancurculio and kill them? And the third one was to determine the efficacy of Sternenema riobravi, Carpocapsi, feltii, and Scarabei to control plancurculio in high bush blueberry fields. Okay? So I'm going to real quickly in the methodology here. So for the nematode persistence in acidic blueberry soil, we used cups filled with 100 grams of moist acidic blueberry soil, and we applied 200 IJs of Sternenema carpocapsi, feltii, Rio Brave, and Etorhabditis bacteriophora. And to check the nematode persistence at different times, we checked these cups at 0, 7, 14, 28, and 56 days after treatment. And we added 10 wax moth larvae as baits for the nematodes. And every 3 days, dead wax moths were replaced with live ones, and baiting was concluded 12 days. So it was a 4 baiting rounds. So here, it's possible to see in this graph that the infected wax worms was highest in Sternenema Rio Brave and lowest in the Ectorhabditis bacteriophora. The first infection decrease of Ectorhabditis bacteriophora was at 7 days after treatment, and the lowest infectious already at 28 days after treatment. And in the Sternenema species, a decrease was observed in all 3 species at 28 days. However, only Sternenema Rio Brave further declined by 56 days after treatment. Okay, so we already have one answer for our objective. Okay? For the second one, the virulence of EPNs to Plancurculio larvae. So we used basically the same methodology. The cups with soil, we used 5 Plancurculium fourth insta larvae that were added as baits for the EPNs, and 4 EPNs were tested. Sternenema carpocapsi, feltii, Rio Brave, and Scarabei. And they were applied to the substrate surface at 3 different rates, 100, 200, and 400 IJs per larvae. So we performed the evaluation. I performed the evaluation at 7 and 14 days after treatment. And here we can see that at 7 and 14 days, Sternenema Rio Brave caused the highest Plancurculio larvae mortality at low, medium, and high rates, followed by Sternenema carpocapsi, Sternenema feltii, and Sternenema scarabei. So to test the virulence of these EPNs to Plancurculio pupae, I used the same methodology, however, it was small cups, so it was a solo cup, plastic cups that were filled with 20 grams of moist acidic blueberry soil. And to determine the nematode virulence, we added 1 Plancurculio pupae as bait for the EPNs, and we tested Sternenema carpocapsi, feltii, scarabei, and Etorhabditis bacteriophora at a high rate, 400 IJs per pupae, based on the data from the previous experiment. And Sternenema Rio Brave were tested at 3 rates, 100, 200, and 400 IJs per pupae, the same ones from the previous experiment. Okay? So we wanted to see if these rates for Sternenema Rio Brave will also infected the pupae. Okay, so here we can see the graphs for 7 and 14 days at 400 at a higher rate, 400 IJs. And at 7 days, no difference among treatments. There was no difference among the treatments. And at 14 days, there was a highest plancurcular pupil mortality was detected for Sterninema carpocapsi. It was a highest mortality, but was 50%. And here we have for Stenenema Rio Brave, where we tested 3 rates, a low, medium, and high rates. We can see that at 7 and 14 days, 7 days is black, 40 days is gray. We have a highest C. plancurculi pupae mortality was detected for Stenenema Rio Brave at high rate, 99%, and median rate, 97%. I would say that the low rate was about 85%. So based on the results that we have in the laboratory, and we have a really positive results, so we decide to go to test this in the field. So the field experiment, it was performed in the blueberry site at Philip Marouk Center for Blueberry and Cranberry Research and Extension of Rutgers University. And 4 EPNs were tested, the best one for the lab. Scarabei, Rio Brave, Carpocapsi, and Feltii, plus a control treatment. So for this experiment, 100 blueberry-infested fruits, they were distributed on the soil surface of each of 24 areas. So here in the first picture here, we can see the blueberry fruits on the soil surface. And these blueberry fruits, they were infected with plancurculio larvae. The areas were treated with 50 IJs per square centimeters of a given EPN species, and it was applied by a hand sprayer. After the applications, the plots were watering and emergence cages were placed, as here is the picture in the middle. So to check the nematode efficacy, the cages were inspected twice a weekly for 6 weeks, and any plancurculio adult emergents were collected. So the data from this experiment in 2020 showed that Sterninema rio brave provided significantly higher levels of plancurculio suppression, 90%, than the others entomopathogenic nematodes. In 2021, we repeat this experiment in the field, but we change some things in the methodology, and we tested only Sterninema rio brave at low and high rates, plus untreated control. And instead of using infested blueberry fruits, we infested the soil surface with 50 plancurculio larvae fourth insert that they will burrow in the soil to turn pupae of each of 21 areas. So the areas were treated with 25 and 50 IJs per square centimeters. This was also applied by hand sprayer. And after the EPN applications, the plots were watering and an emergence cage was placed. So here is the overview of this experiment. And to check the nematode efficacy, the cage was inspected twice weekly for 6 weeks, and any plancurculio adult emerger was collected. So for the results in 2021, we can see a significant difference was detected in the plancurculio adults recovery comparing to control and Sterninema rio brave at low and high rates. So there is no difference between the Sternenema Rio Bravo rates were detected, and both rates provided greatest level of Plancurculio suppression. I would say that the high rate provided 100% of suppression. What are the conclusions of this research? So Sternenema Rio Bravo is the EPN species that better persists in the acidic blueberry soil, is the most virulent EPN species against Plancurculio larvae and pupae, and it is far the most effective EPN species to suppress Plancurculio larvae and pupae in high bush blueberry field. So what are the outcomes for this research? So we presented this research in the extension presentations. We showed the data. We showed to the growers how easy is this data, because they don't this research. They don't need to place the cages there. They don't just need to apply the Sternenema Rio Bravo. So biological control using entomopathogenic nematode is being implemented in organic blueberry farms to control Plancurculio. So yes, we helped the growers, the blueberry growers, to overcome one of the challenges related to um, insect pest management. However, there are still challenges in controlling other insect pests, such as Drosophila suzukii, the spotted winged Drosophila, in both organic and conventional farms. So this is the last slide. I'm almost done. So it's just an overview regarding the major, current major challenging in controlling insect pests in blueberry farms here in the United States is to control spotted winged Drosophila for organic and conventional farms. Why? First, the Drosophila activities. They start in May and go until late September. They overwinter. The adults overwinter, of course, during the winter season, and they seek shelter in the decaying fruits, husks, and skins. So after harvesting, the growers, they have to make sure that the field is cleaned for all fruits that have fallen on the ground. The field needs to be clean, because if the field is not clean, the adults will overwinter in these fruits, in the skins. The Drosophila suzuki can cause devastating losses in blueberry productions, with the potential to render entire crops unmarketable if left unmanaged. The pest's ability to attack ripening fruits make it particularly challenging to control. And also, they have more than one, they have various generations in a year, right? Remember that I told that? Economic impact extend beyond direct crop losses to include increased management costs, potential market rejections, and long-term changes in production practices. So Drosophila suzuki has forced many growers to adopt more intensive pest management strategies, often increasing reliance on insecticides. So this comes where, at the same time that we are having problems with Plancurculio, we also are having problems with Drosophila suzuki. So, and the blueberries, they need the bees to pollinate. So when they apply the insecticides, they can kill the bees. And also, certain insecticides, if they're applying nematodes, certain insecticide can affect the entomopathogenic nematodes on the soil. So it's been a huge challenge. Organic producers face particular significant challenges in managing Drosophila Suzuki while maintaining certification standards. Um, Organic producers, they cannot use insecticide at all. So, and ongoing research is crucial for developing sustainable management strategies, including improved monitoring, biological controls, and resistance cultivar. And this ongoing research, it's um, it's involved the relation between the growers and the extension programs that we have in the universities. So this is all for today. And this is all that I wanted to share with you about blueberry farms here in the United States. Um, I hope you guys like it, and I'm open to answer any questions. Thank you so much.
So, guys, any questions? Oh, we have already one, Nana. Let me see if you can see it. Mm. Can you read it? Are you?
Yes, we already see a few blueberry farms in
Yes.
Brazil, but are there researchers about blueberry breeding for its adaptations in hotter climates? That's a good question. Probably yes, because since we have some blueberry farms in Brazil, probably the breedings are working on this adaptation for hotter climates.
Karina also has a question.
Thank you, Karina. Can you tell me if this research can be applicable here in Brazil in mirtilo crop? You mean regarding the if you mean regarding the entomopathogenic nematodes? I would say that first we have to check how these nematodes will have the virulence in the soil, because for the blueberries we have acidic soils and we see that they work, but I'm not saying regarding the pH, because I know and the soil type, because the soil type here is completely different. That was another option that we see. So I would say yes, but we would need a further investigation before applying these nematodes in the field.
So, and if you had a little more time, we have some more three questions. It's okay?
Okay. Thanks, Efranio. How is the adoption of biological products today in the United States by the farmers? They are receptive to these new tools? Oh yes, a lot. As I mentioned in the presentation, the growers here, they have an open mind, and here they really care about uh, the regulatory compliance regarding the insecticides. So they are really looking for new tool to use in the field and do not use insecticides anymore or reduce the use of insecticide. So yes, they are open for the biological control here in the United States. But Vinicius, I have a questions about the average shelf life of blueberry, both at low temperatures and ambient conditions. Thanks again. Um, so these are really good questions. I don't have exactly a shelf life time for to answer your question, but the only thing that I know is that as soon as we get the fruits from the field and they go to they they get the fruits as in a bucket, and they cannot fill this bucket because if they fill this bucket can be too heavy and they can have a damage in the fruits. So there's a lot of things that we have to consider. So they harvest, they hand harvest the fruits, and then they take to the storage where they're gonna um, split the amount of the blueberries and there was in the bucket in a small containers, and then they will keep in this the a cold room. I would say a cold room uh, to um, to keep a quality of the fruits. But I don't have a shelf lifetime to to give to you.
The last one, it's from Fronio.
Yes, Afranio. Here in the United States, I would say that is not uh, easier, but it's much more faster, and we have much more companies with that have um, more options that provide more options for you. For example, I don't know for now, honestly, in Brazil, but since I was living in Brazil, we have some companies where they have only, for example, predatory mites and BT in Bavaria to sell to the farmers, and they didn't have other products. And here in the United States, first we have more companies, um, and second that we have much more options available, and the register uh, uh, to a biological product here is much more faster than in Brazil.
So I think this is it, Anna. Thank you very much for your time and for being here and sharing all the research. It's really interesting and it has a lot to everyone to learn a new topic here. So we are going to have a break now, 10 minutes, and we are going to go back at 7:20. Okay, guys?
Thank you,
And thank you,
everybody.
Anna,
Thank you,
again.
Yuki. Bye.
Hi guys. So we already have our time break. We are going to start from the second speaker. So our second speaker is going to be Victor Augusto, and I'm going to give a little introduction. Victor is an undergrad student in animal science in the Federal University of Lavras. He completed a research internship in Virginia Tech, participating in research at the University of Florida, and it's a researcher member in INPAR. He will address the impacts of efficiency on sustainability in dairy farm, and we are eager to hear about the efficiency and the dairy farming. Victor, the floor is yours.
All right, thank you. Can you hear me well?
Yes,
All right, good. And can you see my screen as well?
yes,
All
we
right,
are seeing
good.
you.
All right, so good evening, everyone. Today I'm gonna talk a little bit about the impact of efficiency on sustainability in dairy farming. So Yuki already introduced me a little bit, but I prepared a slide for myself. So my name is Victor Augusto, like I said. Originally, I'm from Mogi das Cruzes, São Paulo, and as a personal thing, I was always passionate about dairy cows since I was a little kid, and when I decided to go to the university, I went straight to the dairy. So I started animal science because of cows, basically. So in 2019, I entered at Federal University of Lavras in the animal science course. And since the first semester, my first year, I started working in the UFLA with an extension group that also works in the dairy farm of the university. So I was able to have a lot of hands-on experience, a lot of field experience since then until now. So I'm currently in the affiliate until today. And in 2021, I entered the import lab, which is the Dr. Marina Danis lab, focused on dairy nutrition research. When I found out my second passion, professionally speaking, which is research. So basically, I'm passionate about dairy cows and research focused with dairy nutrition, right? So, like Yuki said, last year I had a international experience in the University of Florida in Virginia Tech for 1 year, 6 months each. So, mainly focused on dairy nutrition research, right? In lab, in the field, in the farm. So let's start. Today I have three main objectives for the presentation. So the first one is to show you guys like the currently situation of our country in the carbon footprint when you talk about dairy industry, right? Show you tools to achieve efficiency and inspiring you guys somehow, everyone who is watching, either on the sustainability, production, efficiency subject, either like as a person, like a student, and everything related to that. All right. So the presentation, I want to answer this question, like how production efficiency can bring sustainability to the dairy industry. Because sustainability is something that is like is a trend, all right, for the next 50 years. So every system, every industry wants and have to be sustainable, all right, for even like social economics, but for the real thing, like we have to do it because uh, if not, you're gonna over with our planet, right? So that's the main question that I want to answer it. So at first, where are we? So where is Brazil? Where is our country in the carbon footprint, right? So we have three stages. I didn't bring the like the quantity of carbon emission and everything. It's not the objective here, but basically we have three stages, three levels. So the high carbon footprint level, the medium, and the low. So the high is like the worst, like the high is the worst, and the low is the better. Like if you produce less carbon, is you're more sustainable, and consequently, like you're more efficient as well. So we are at the third one, right? So we are a high carbon footprint level when we're talking about the dairy. All right. So I'm gonna each level has a characteristic. So when you are in the lowest level, you have like the most opportunities to do something about it. You won't have a lot of investments to do it, and you can do it something like it's more easy, like it's easier to do it when you are in a medium or a low level. Because when you're in a low, you have to buy additives, you have to buy a lot of like expensive technologies. All right. So we are in the high and have more opportunities, and that's good. All right. Because when you have more opportunities, these opportunities are basic adjustments. So when you do it, these basic adjustments, you have a payback. Like it's easier to get a payback. All right. And when you have a payback, you have profit, and you can end the cycle going to the next level. If you sustain that, like the adjustments, you sustain the adjustments, you're gonna reach the next level, and you're gonna have another opportunities, right? Another strategy that you're gonna go to the low. That is the goal, all right, for every sector. So today I'm gonna focus my presentation in the basic adjustments, and my whole presentation is about that. So which adjustments we can do it to get out from the high level to the medium, all right? That is a big movement. So we can manage the herd structure, the genetics, nutritional. I put in green here because it's like the main thing I'm gonna talk about. Reproduction, health management, waste management, um, agriculture planning, which is really important because you have to, if you produce milk, you have to be a good agriculture producer as well, because you have to fabricate the food you're feeding your cows, all right? And if you do a bad food, like a bad corn silage, a bad concentrate, the cows won't respond. Like they will produce less than you want them to do it, all right? So this is really crucial as well. And a better animal welfare. This last one is the least, but not the less important, because it's really, really important. Because if you do all these adjustments, like herd structure, genetics, nutritional, waste, everything, if you don't have a good welfare for the cows, they won't respond. All right. This is another whole presentation, so I have just one slide for animal welfare, and this is a image like for the trend that like I said a little bit earlier in the presentation. The trend is to sustainability, like reducing methane, and for that you have to improve the welfare and the use of technology. And my presentation will mainly focus on the use of technology, and I'm gonna talk a little bit about improving welfare. So we're gonna get these two tickets and continue the presentation. So for animal welfare, you have to treat your cow like I don't know, maybe like a princess, something like that, like in a spa. All right, because if you don't do it, she won't respond. She won't produce the milk they want her to produce it. So we have to have good nutrition. You have to put like the cow in an environment that she can express her behavior, like the most proximally behavior as natural as possible, right? And with that, you have a better health, older health, food health, everything. And with all that together, the cow will have a mental welfare, like a good mental welfare is crucial. Like for her, she has to be calm, she has to be in a quietly, not heat stress environment and everything. So this is crucial. Like everything that I'm gonna say in the next slides, if you don't do it this, you can't, like, you won't achieve what you want. That's the thing. All right. So let's start. First, you have to put a target, like what do you want to do, like what you want to achieve. All right. So I'm going right now. My farm is producing 24 kilograms of milk per day. I want to produce 35. All right, I have to put that target. But how am I gonna get to the target? All right, you're gonna have to make the adjustments and be consistent. Have consistency. All right, have to have discipline. Have to do the same thing, the right thing every day, like every day, everywhere, like always. All right, and we have to do that with excellence. So if not, it's gonna be like, you can be mild, you can be so with if you want to achieve something in the dairy system, like, because everything is like crossed together, like everything's like crossing over, like the reproduction, the production, the feed, the agriculture, the waste management, like the environmental, like bad, that like the cow, the bed you're putting the cow, if it's not good, she's gonna have health problems in the udder, mastitis. She's produce more milk. You're gonna have more cost. Have to apply antibiotics. You're not gonna have have a sustainable system if you're applying too much antibiotics. All right, you're gonna have losing money, because when you apply the antibiotics, you can send the milk to the industry. So you have to waste that, you have to dump the milk. So we gotta do it with excellence. All right. So with that, you be achieving efficiency. All right, and efficiency equals to less waste. So if your waste is low, you are efficient consequently. All right. So if you're wasting less food, you're wasting less milk, you are being efficient. Right. It's something really relatable. Like they're in the same quadrant, I can say. Yeah. So one of the tools that we can use it to achieve the efficiency of the production is the Six Sigma. Is like tool really common use in companies, in the industry, but you can use it in the farm as well, and you can use it right. So you have some stages. So at first, you have to define, like I said in the last slide, you have to define, have to put a target, all right? So define the goals, define what you want, from where you are. So where do you want to go, right? But in order to define the goals, you have to measure. This is one of the most important stages. Like if you don't measure, you don't know where you are, right? And if you don't know where you are, you can know, I want to go there, but you don't know where you are exactly. So we have to measure. This is essential. All right. So measure everything possible in the farm: milk production, reproduction, then like the feedback, everything you can. All right. And the next thing, when after you measure everything they can, everything possible, you analyze that. So you're gonna analyze the data, and with that data analyzed, you're gonna create indexes, indicators for the farm. So like how I know how much I'm producing, I know how many cows I have, the reproduction status of each cow. So you're gonna have like everything like playing out in a sheet, and you're gonna have it in the computer, like you can see even the paper. You can just know, like if it's a small farm, you can just note. All right, you don't need a computer exactly or a whole software to measure and analyze everything. And then after that, okay, I know what I want to go. I know where I am right now, and I analyze everything. And then I jump to the improvement. All right. So I know what I have to do. I know what I want to go, where I want to go, right? And I have to make improvements to reach that stage, that goal. So improve the process. So we have to improve all the process. All right. So if you want to improve the milk, you have to improve the feeding, the milking parlor, the environment, everything. So Then you jump to the improvement process. And after that, you have to control it. All right. So if you don't control it, if you don't have the consistency, you won't, you can achieve that. But like as soon as you see, you're going to be like low again. You're going to be like out of your goal. So you have to control the improvements that you make. You have to be consistent. All right. So here's a phrase from Midger and Peters from 2010, and the presentation, the Six Sigma, is based on this phrase. So precision, related to the dairy farming, right? To a farm. So precision feeding is an information and technology-based management system to identify, like measure, identify, analyze, and control variability in farm process to optimize performance, profitability, and sustainability. All right. So I'm gonna focus right now on the next slides on this phrase. And right now I'm gonna talk about more technology, because when you have technology associated with the process, you can gather more information, right? And nowadays we have a lot of information, like a lot. You can have like photo shoots for the back of the cow, so you can have the body scar condition. You have cameras to evaluate the behavior of the cow, the calves, everything. You have collars to monitor the activity of the cow. So you have a lot of technology, right? So the thing is how you're gonna use the data, right? How you're gonna apply the information that you gathered. So this is just a brief picture of how is it going for the technology. It's not that updated. It's like the last data is from 2017. I guess right now we have a lot more studies than that, like a lot. I don't remember exactly, but more than 100 studies per year, all right, for the technology. This is a data from a professor from Wisconsin, Madison, and here's a graph of like the which activity or which category the technology can measure, so you can use the data. And the most used one, like 75%, is from estrus, like reproduction status. Lameness is like the behavior of the cow, and mastitis, that is a big problem in the industry, right, in the farm. And the other interesting is, and most of this, all this technology, the research, the studies comes from the dairy. All right, when you can see in the left here in the bottom, the dairy cattle studies like are responsible for 92%. When you talk about cattle like dairy and beef, the dairy is more than 90%. All right, and I think it continues like that nowadays. So we have a lot of things. So we have automatic feed bunks. This is a product from like that is from Brazil, Intervalo, that can measure the intake of the cow. So we have a lot of things related to feeding. This is a graph of studies made with Intervalo throughout the years. So in 2020 and 12, we have 24, and 4 years later, jumped to 180. All right, and so it's something that in Brazil is not just in the U.S. All right, it's in Brazil have a lot of technology as well. So we have like facial recognition. We have a lot of hackathons, like movements that make the ideas to startups, to companies. And so we're in a good path when we talk about technology here in Brazil. And now, when we have all this technology, we have information. But like I said, we have to you measure, you have the information, but how are you gonna use it? All right. So this is the main thing. How are you gonna use the information so we can have more efficiency? You can do whatever you have to do to achieve your goals. So first, this image here is really nice to see, because it's more you can picture. All right. So you have the data, like the raw data that you can gather, and that data you transform into information, like the milk production of each cow, the reproduction status. Like you transform the information. Information is something that you can take something from that. All right. So and then information can transform into knowledge. Is when like you can associate like the milk production of the cow with the reproduction status. So you build knowledge, and knowledge is really useful in a farm, right? You have to have the knowledge how the cows are going, all right? How the cows are producing, how they're feeling, all right? So the knowledge is the main thing, all right? So let's jump to the feeding, all right? We have the information, but we have the information from the cow, information from the nutrients that we're gonna offer the cow, all right? The info that we put is gonna be the input. We're gonna formulate the diet. We have like to monitor it, all of the management for the feed bank, the silos, the mix wagon, and everything. So it's a whole system to feed the cows, right? So you have to. The cow won't give milk for you if you don't feed her right. So this is it's crucial, and also it's like 50 to 65% of the cost of the operational cost. So it's really important to be efficient here, because it's responsible for 50-60% of your cost. So if you waste here, you're going to be losing money, and consequently you're not going to be sustainable, because it's going to have so much waste, right? So this is like the main thing of dairy nutrition, of dairy farming, right? The feeding is really, really important. So the next slide is going to focus on the cow in the nutrients, all right, and in the formulation. So to formulate a diet, you have the nutrients, like what I can offer the cow. Like you can offer like fiber, starch, energy, like protein with soybean meal. I need to form corn silage, a pasture, a forage. What I can give you to her, and you have the requirement, like what the cow needs to produce a certain amount of milk, right? So we have to balance that, like what you're gonna offer and what she needs. So this is the basic thing of diet formulation for dairy cows, all right? So right now, I'm gonna focus about the nutrients. So it's the same thing as the Six Sigma, like that I said in the last slides. You formulate a diet. You need to know what exactly you're gonna feed. Or you need to know what exactly you're going to feed the cow, right? So you need to know the feed composition. You need to know like what you're gonna give in the cow, all right? Because feeding, like the feed, it varies a lot, right? So this is it's just an example, like a variation of NDF in a corn silage. So look at the variation. That is a lot. So imagine one day you offer that, you formulate the diet, and you think it's like that, right? And then the other days like that, other days like that, other days like that, right? So you have to know what exactly you are feeding your cow. And for that, right now we have a technology. We have NIRS, all right, a near infrared spectroscopy, that it just get the sample sent to the lab. It's a little expensive, like nowadays is around 300 reais per analysis, but it will you have like the results in 2 days, maybe even less than that, and you need that. You need the information to formulate. If you don't do that, you're gonna formulate in the like you don't know what you're doing, right? Because you can formulate for 35 of NDF of fiber, but actually the forage has 40. Like it's completely wrong, right? And I also have the NRS you can bring to the farm, so it's more expensive, but it's way faster. Like you can adjust the diet whatever you want, right? So you can get the feed, analyze it, like in seconds you have the results, all right? So the thing is, you have to analyze it, right? But if you don't sample it right, like you can't do anything as well. You're gonna do everything wrong, right? So here's a work where they catch just a sample of each spot here. So here in the black spots, like here 27, 23. Each spot is a sample that they analyze for starch level in the corn silage. All right. So look at the variation here. We have a point that we have a 42% of start and a point with 14. That is a lot of variation. A lot. 26, 30, 23. So if you just go to the silo, to the panel, and get just one point, like I go to the silo and I grab like 200, 300 grams of samples, like of sample of corn silage, and I get just from one point. I don't get like one from here, one from here, one from here. I just get one. So I sent to the lab and I got back the results. I got 26. But actually, like when I feed the cow, the starch level of the silage is around like 35. So you're formulating for 26, but it's actually 35. So you're overfeeding your cows by a lot. So that is, the sampling is crucial, so you can have the right result. So it doesn't matter if you have information, but you gathered the information, the data in the wrong way. All right. So you have to follow the steps with excellence, all the steps. So like if you formulate like for 42, but you put like 14, I don't know, if you like I example earlier, a silage of 26, but actually it's 35. All right. So this is another experiment where is a professor from the Ohio State University, Dr. Wise, where they gather a lot of samples analysis. All right. And they found out that 36% of the variation in the NDF content of corn silage was due sampling. So if you sample it wrong, you're not gonna have a realistic result. Right. So now I talk about the nutrients. Right. You have to know what are you feeding the cows, and the other way of the balance, you have to know what the cow needs. All right. So this is the other crucial part of the balance. What the cow needs. Which are the animal requirements for the cows. All right. So in order to know the requirements, guess what? You have to measure it, right? You have to measure it. You have to measure the milk production is the first thing. Like if you can't do anything, measure how much your cows are producing. Because if you're not, you're formulating for a cow you think that your cow are producing 15 kilograms of milk per day, but they're actually producing 25. So you're subfeeding your cows, right? They need a feed to 25, but you're feeding them to a feed to 15. So they're gonna stay to the 15 or even lower than that, all right? Because they're gonna feel it. They're gonna be hungry, all right? So milk production is a measurement that you have to do it in a dairy farm, or you're obligated to do it, all right? Milk analysis is a little bit expensive, not that expensive, but it's crucial as well because a lot of input data that you have to put in the software when you're formulating a diet, you have to know the protein and fat content of the milk, because it's the content of the solids of the milk drives the requirement of the cow for energy, for protein and everything. So you have to know how much of protein she's producing, how much of fat she's producing, so you can give the right amount of energy, the right amount of amino acids and protein, right? And this one here I put it in red because it's something really important, crucial as well. You have to weight your cows, how much they're weighing, all right? Because if not, is another data that you have to input in the software that is crucial to determine the dry matter intake of the cow, the production. So it's something that is crucial, but not many farms do it, even like big farms, because it's a big problem actually, because it's simple, like just wait it, but actually it's not that simple to do it, because there's not management to do it. If a lot of cows is not good, you have to take all the cows in a day, and weigh them is not doable actually, right? But you have the technology to put a scale like after the cow is getting out from the milk parlor and getting back for the beds. You can put like in a way, and they're gonna measure like every day, but it's expensive, right? Here in Brazil, it's really expensive. Back in the United States, a lot of farms has the scale after the milking parlor. But here is something really difficult because if you formulate a diet for a cow that has, you think your cows like just looking at them that your cows have 700 kilograms of milk of body weight, sorry, but actually they have 600 kilograms of body weight, right? So you're formulated for 700, but your cows actually have 600. So you're gonna overfeed them again, and with that, you would have waste, and with that, no efficiency, no sustainability, right? Another thing is calving date. So we can plan the when you're gonna dry off the cow, when you're gonna change the diet. So the cow, like, there's a stage of the lactation that the cows starting to produce less, and if you maintain that in a pen that is with the cows that produce more milk, she's gonna eat more than she needs, and with that, she's gonna get fat. And when she get fat, she's gonna like be in the next lactation, she's gonna have like metabolic disorders, more problems, and consequently produce more less milk, right? So this is four main things that you have to measure to know what your cow is requiring, like to respond to a certain amount of milk production. So another thing that you have to do, and you must do it, but again, a lot of farms don't do it, right? Is grouping your cows. So you have to group your cows accordingly. Like each cow, like the less variation, the better, right? So if you have like a pen with like this orange cow with the blue cow and the green cow, the variation here is gonna be like way too big, right? And you're gonna have a lot of situations. You're gonna overfeed some group of cow, subfeed another group of cows, and with that waste again. All right. So I have to group the cows, have to block the cows, right? So put the cows with a similar milk production, a similar days in milk, similar lactation like primiparous and multiparous cows. All right. So when you do the grouping well, you have a less pen variation, all right, within the animals, and you meet the requirements correctly. So that's what you want when you're talking about formulating a diet, to meet the requirements. All right. So here's an example. So I have this group of cows in the like above here, like all together, and you need to group them because this group here, when you do that, because whenever you're formulating a diet, you formulate to a pen. You can't formulate a diet to an individual being, right? You can't formulate a diet to an individual cow because she's going to be in the pen eating with all the cows together. So what we do when you formulate a diet is to formulate the diet to the average. All right. So the average of this pen here, with all these cows with different stage of lactations, different productions, is 35 kilograms of milk per day. And the days in milk is like the days that the cow are producing milk. So she calved, and we start counting the milking the days now, right? So it's 145 days, the average. All right. But when we see each group, the cows like more closer, is way different from the average here. For example, you have like the red one here. The average of milk is 30 liters per day, and the days in milk are 50. So early lactation cows. The orange is like high producing cows, 50. The green 38, and the blue ones here 21 liters. So if you formulate for 35, these cows, the orange cow is gonna be like sub-feeding these cows because they need feed for 50 liters, all right? But you're just gonna give them 35. So with that, the production of this cow is gonna be, they're gonna go down, right? If you don't adjust it. In the opposite, these blue cows here, if you feed a diet for 35, you're gonna be overfeeding these cows. So like I said, they're gonna get fat, gonna have metabolic disorders in the next lactation. So this is really essential that a lot of farms don't do it well. Like they do it, they group the cows, primipros, multipros sometimes, but they don't do it as they had to do it, right? So this is something really crucial that is simple. Like you don't need technology, you just need to measure it. And if you do that, if you group your cows, you're gonna be more efficient, like for certain, all right? And thus you're gonna have more sustainability in your system. So another thing that is bringing some efficiency to the feeding is the automation, all right? So like for example, robot milking, automatic milk systems. We actually have one in the dairy farm of our university at UFLA, and it's really interesting because you have so much data that is actually overwhelming because there's a lot of data that it can gather. And the thing is, what you're gonna do with that data. But when you have something like that, it's easier to get data. All right, so you just have to do it. Also, we have milk analysis in the milk parlor, so you don't have to send the milk to a lab to analyze for protein and fat. You can measure it every day. So this is a really good technology. This AF milk. Also, we can put a scale in the water fountain. So whenever the cow is gonna go to drink water, you're gonna measure her body weight like automatically. So these are more expensive, like technology, but it's gonna be something really common in the next couple of years, like the cell phone. Like back in the days was like a touch cell phone screens was really like a new thing. Not everyone had, but 15, 10 years later, everyone has one, right? So this is are the trends for the automation in the dairy. All right, so let's go back to the feeding again. I just explained about the formulation of the diet, so the nutrients, the requirements, the formulation. And right now I'm gonna jump to the feeding. All right, so how we actually feed those cows. So you have like the formulated diet versus the fat diet. All right, so you can formulate a good diet, but if you don't manage well the mix wagon, like the collaborators, the silos, you won't have you won't give your cows the same thing you formulated, right? So you have to measure the dry matter intake at least one day per week for the corn silage, for the forage that you're offering to the cow. You have to analyze, send to the lab, like to analyze the feed composition at least 2 times per month, and also the day-to-day basis. You have to be precise when you're loading the wagon, all right? Because you, for example, are formulating, so we have to put 15 kilograms of corn silage, 5 kilograms of soybean meal, and 2 kilograms of haylage, all right? But your collaborator is putting like 20 kilograms of corn silage in one day, 10 in another, 80 kilograms of soybean meal in one, 7 in the other, 4 kilograms of haylage in one, 3 in the other. So it varies a lot, right? When you have that variation, you will prejudice the dry matter intake for the cows. You're gonna be wasting the feed because you're gonna be overfeeding or subfeeding. So when you're subfeeding, you're gonna produce less milk in that day. And the cow, sometimes when they drop the milk production, they don't go back as fast as you want. All right, so you're gonna be losing money as well and waste your feed and the milk as well. So there are technologies that can measure the precision of the loading. All right. So like above 90% is acceptable, is good. Like above 85 to 90% is really good. So like if you're putting 10, you need to put 10. The collaborator is putting 90.5 kilograms of corn silage, right? So this is something that you have to, when you have a mixed wagon, you have to monitor it every day. All right, and also, all right, you fed, you mix all the nutrients together, the TMR, the corn silage, the concentrate, everything, and have the TMR, the total mix ration, right? But you have to audit that at least at some point, right? To see how is it going. So we have here is like the methodology. You have the feed bank. Here the cows are here and they eat here, right? So you get samples from a lot of points throughout the feed bank, and you do a evaluation in this pen state here. Then have a methodology to shake and everything. And when you have a variation of 1 to 4% is excellent. Like you're doing a good mixing of the TMR. 5 to 8 is good, and less and more than 10% is a problem. Like if you are more than 10%, you have to investigate that. All right. So you don't want, for example, in this, the pen has a lot of levels. So you measure the quantity of each level, you weight, all right? So you mix it, put the sample there, mix it, and weight how much of the TMR are in each level of the pen here. So you don't want, for example, in the bottom pen here, you don't want like 10% here, 15 here, 32 here, 20 here. That is a lot of variation, right? So if you have that, the mixed wagons, there's something wrong in the process. You have to investigate that. So there's another thing that is really important to measure in the TMR is the nutrients variation, because you have to get a sample for the TMR as well, send to the lab to analyze, because when you don't mix it together, like this data here from Dr. Olberg in 2011, when you overfilled the feed wagon, the variation is way higher when you fill the mixed wagon correctly, right? So it's really important to do the process right. And another data from Dr. De Vries in 2014 is a professor from University of Florida. When you have a nutrient variation of less than 5%, you're gonna produce 1.2 more milk, 1.2 kilograms of milk per day, right? So if you vary more the nutrient within the TMR, you're gonna produce more milk because it's more consistent for the cow. So now I'm gonna just focus on the cow and the mix wagon. Like this is the other process, other line of thinking when you have all the feeding. So we have, like I said, have the formulated diet, that is a fed diet. And one thing that is really important to measure it is the intake. So you put a feed in the diet, you put in the cows, but you need to know how much your cows are eating, all right? If not, you can check if you're formulating right, all right? So this is something that is simple, again, but some farms don't do it as well because it takes a little bit of work, like extra work to do it, all right? But it's something really important to do it because you have to check if the cows are eating the right amount of food, and if not, you have to adjust it, right? So this is critical to the dairy farm. You have to evaluate, you have to measure it, the intake of the cows. So it doesn't matter if you formulate, and if you don't measure the intake, you're gonna have problem, all right? So you're gonna have a waste, gonna have less profit and everything. Like I said, you're gonna have waste because, for example, if you feed a quantity and formulate for that quantity, but the cows they cannot eat like the quantity that they put, so they're gonna be a lot of leftovers, all right? So that's a big problem. Like we're gonna just dump the leftovers, And if you're subfeeding them, that's another problem as well, because you have less waste, like not like none. You don't have a waste, but your cows are hungry, like you're not feeding them right, so they're not going to produce well. So there's the intake is something that you have to monitor every day. So the better the grouping, the importance of grouping the cows right. The better the grouping, the better the precision of your dry matter intake data. All right. So for example, if you have a group here that has a cow, like I said, in the blue, in the example that I said, and in the red, in the green, like cows with different body weights, different stages of lactations. These cows eat different from that one, and from that one. So you have the average of the intake, but the variation within the cow is going to be way higher. So you don't want that, right? So here I'm just going to show some real quick examples of indirect indicators. So if you don't want to have the extra work to measure, to weight the intake of the cows, you can have some indirect indicators that you can just look and get an idea what is happening. All right. So you can evaluate the odds, the leftovers of the cow by looking. So we have less, or when you have experience, you can tell by looking. It's like at some point, like it's not precise, but you can at least see if the cows are being overfed or overfed or subfed, right? Feces valuation. So it's something that you want to look at. So here, for example, in this photo, we have a good feces. Like in the second photo here is a cow with an acidosis. So you probably are feeding too much concentrate for the cow, for the pen. So this is not healthy. So it's something that you might, even if you measure the intake by weighting, looking at the feces is something that you must do it. If you wanna be a good nutritionist, you must at least see it when you go to the farm, right? Another one is if you go into pastures systems, the residue of the pasture, like if you have enough food for the cows, because if the cows not gonna eat these, all right? So they eat everything available here. So you don't know if it's enough or not. Probably not, because if you have more forage here, probably they would eat. All right. So this is something that we can look when you're looking at pasture systems. Rumen score. So we can look at the left side of the cow to see if the rumen is filled or not. Like this cow is like it's not filled enough. This one as well. So you can say by that the cow is not eating. All right. So it's something that we can look at the cows, right? Just look at the cows. You can get something for them every day. So that is efficiency as well. All right. You don't have to have a lot of technology. Just look at the cows. And there's other indicators that requires more technology, like the milk analysis. You can, if the cows have a depression in the fat content, it means that you're giving too much energy, too much concentrate, too much starch, and the rumen is not working well. All right. So you can look at that, the protein production as well. Maybe you're missing some protein in the diet, some amino acid. When you have like a fat lower than the protein, you have a big problem there, because usually the fat is way higher, like 3.7, and the protein is 3.10, 3.2. So if you have an inversion of that, bad relationship, there's something wrong with the diet. All right. So this is a measurement, is a data that you can look at and figure out if there's something wrong within the diet. This is like PDR, crude protein relation with like nutritional status of the rumen, and rumen degradable and degradable protein. You can measure the milk, like in the parlor. The ration balance, like the feed. If you change a feed, for example, I'm putting corn silage and I'm gonna put like wheat silage. Like look at the fat here in the yellow, it's low. because it might be something wrong with the feeding, so we have to investigate that, right? So to conclude my presentation, the mentality. So we have to have a mentality to change it, right? To be efficient. So changing is uncomfortable, but if you want to achieve something, it's necessary, right? So and if you keep doing things the same way, we always leave you in the same place, and it's bad and good, right? Because if you are consistent doing something wrong, you're gonna be there forever. But if you are consistently doing something right, you're gonna be there like for a long time. I don't know if forever, but it's gonna be there for a long time, and that's good, right? So it depends on how you're doing your stuff in the farm. So if you get more data, you have more control, more technology. Consequently, you have less variation, less error, and less waste. And what all that means? More efficiency and more sustainability. All right. So if you have all that, you are more efficient and consequently more sustainable. And the thing is, one of the final messages like keep it simple. Like if you can measure, if you can buy a scale, if you can send to feed to the analysis for like every week, send at least to them to like one time per month. The thing is, keep it simple, like rice and beans, right? Just do the simple, the rice and beans with discipline. Be constant, right? Do the simple, be constant, do it with excellence, and you will achieve your efficiency. And here's a phrase from Cole Bryant, like a guy that inspired me a lot. It's great things come from hard work and perseverance, and no excuses. So you have to do it. So no excuses. All right, it's hard, but it's not simple. You have to do it. Just do it. All right, and do it with excellence and constantly. And with that, I'd like to thank you guys for the opportunity to talking to you. I hope you guys could take something from the presentation, right? And I'll be gladly taking some questions. Thank you. All right. No, I can't hear you. I think it's muted. Muted.
Sorry.
Yeah.
Sorry. Thank you very much, Victor. Great presentation. And we are going to go to the questions now. You can see the
All right.
chat.
Yeah. All right. How do you measure things like animal welfare without technology? Because I also agree that it's something that matters a lot, but it's also something that can be really subjective. All right. So that's the thing. The thing is that, like I said in the last one of the last slides, to look at the cows. So when you are experienced, you have a lot of experience. You can tell if the cow is being like treated well or not just by looking at them. Like if the cow is laying down more, that is a measure that you can just look at them, and you don't. And again, and altogether, you don't have to be too much experienced. So you can, if the cows are laying down like a lot, you can tell that they're having a good environment, like a good welfare. So basically looking at them, like if they're not if they're not heat stressed, for example, like breathing like heavily, if they're eating. So that is one of the stages of the animal welfare, the nutrition. So if the cows are not eating enough, if they're not eating like frequently in the feed bank, it might be something wrong with the diet. And again, you can look at the diet and send you to analyze it and everything. So basically by looking at it, by looking at the cows.
Great. So, any more questions, guys? Since we already passed a little bit of the time and we don't have now questions. If we have further, you can send us in the Instagram or something like that. That we chase Victor for the
All right.
answers. So, guys, as the come of the end of the third international webinar on production challenge, I like to extend a heartfelt thank you for each one of you guys to stay participating until now. Your engagement will be the made of the successful of the event. And so a special thank to our incredible speakers, Anna and Victor, for the insightful presentation and for sharing their expertise with us. Your contribution has been valuable, and we are deeply grateful for your time and effort. Thank you very much.
Yeah, thank you guys for the opportunity. It was really nice.
So this is it. Thank you very much, Victor, and see you. So, guys, just to finish the event officially, I hope you have learned some new things and have like the opportunity to connect with field professionals and expertise in the field. I'm going to thank once more for the attendance and make the webinar possible. We look forward to seeing you in the future events. Until then, stay safe, curious, and continue to strive for excellence in your respective fields. And this is it. Thank you very much. And this is the end of the webinar. Bye, guys.